Let's move on to question number 2. Respiration is a process that results in the synthesis of ATP. The ATP can be used within the cell for energy requiring reactions and processes. There are 4 stages in aerobic respiration: glycolysis, the link reaction, the Krebs cycle, and oxidative phosphorylation. Part A. The ATP synthesized in respiration can be used to make larger and more complex biological molecules from smaller molecules. Name the type of reactions that occurs when larger, more complex biological molecules are made from smaller ones. So guys, always remember this point, that whenever smaller molecules join to form larger molecules, or whenever larger complex molecules are made by joining smaller molecules, this type of reaction is known as anabolic reaction. And the reverse of this is catabolic, in which large molecules are cut or broken down into smaller molecules. So catabolic is when large molecules are broken down into smaller molecules, and anabolic reaction is when the smaller molecules are joined to form the larger molecules. Let's move on to part B. The first part of the glycolysis uses ATP. Explain why ATP is needed in the first part of glycolysis. So we discussed this in the video lecture of glycolysis, that ATP is needed in the first part of glycolysis in order to provide activation energy to the glucose so that it can split. Glucose is a very stable molecule, and if you want it to split, you need to provide energy, and that's why ATP is used. ATP is also used to trap glucose inside the cell or to stop the glucose going out of the cell, because glucose can go out of the cell, but glucose phosphate or glucose or fructose 1,6 bisphosphate cannot go out of the cell. So what will we write over here is that number 1 point is that we can write, to energize glucose or to provide activation energy to glucose, we add phosphate to glucose, right? So ATP is used to phosphorylate glucose so that it gets activation energy and becomes less stable, so that it can split because glycolysis is splitting of glucose into 2 molecules of triose phosphate, right? We can also write that ATP is used to phosphorylate glucose so that it's trapped inside the cell or is prevented from leaving the cell, right? Let's move on to part C. State the precise locations of substrate-linked phosphorylation reactions in aerobic respiration. So guys, you need to know that substrate-linked phosphorylation reactions are the reactions used to make ATP, and in aerobic respiration, substrate-linked phosphorylation occurs in glycolysis and in Krebs cycle, right? ATP are made by 2 processes in aerobic respiration. One is substrate-linked phosphorylation, other one is oxidative phosphorylation or chemiosmosis. So we need to state the precise locations of substrate-linked reactions or substrate-linked phosphorylations. So substrate-linked phosphorylation occurs in glycolysis and Krebs cycle. Glycolysis occurs in the cytoplasm, whereas the Krebs cycle occurs in the mitochondrial matrix. So the precise locations for substrate-linked reactions or substrate-linked phosphorylation are cytoplasm and mitochondrial matrix. What happens to pyruvate in the link reaction in aerobic respiration? So guys, as we all know that in the link reaction, pyruvate is decarboxylated, right? It's dehydrogenated. We can also mention decarboxylated to release carbon dioxide, CO2, dehydrogenated to release hydrogens, release hydrogen atoms, which are picked up by NAD to produce reduced NAD. After decarboxylation and dehydrogenation, what happens? After decarboxylation and dehydrogenation, acetyl group is left. Acetyl group, which is of 2 carbon, is left. Which is picked up by coenzyme A to form acetyl coenzyme A. Right. Let's move on to the next part, that is part E. Chemiosmosis is a process that occurs in mitochondria during aerobic respiration and in chloroplasts during photosynthesis. Describe the differences between the process of chemiosmosis in mitochondria and the process of chemiosmosis in chloroplast. So guys, we all know that chemiosmosis occurs both in mitochondria and in chloroplasts. So we need to differentiate between the processes of chemiosmosis in chloroplast and mitochondria, right? So we all know that chemiosmosis in mitochondria occurs during oxidative phosphorylation, whereas chemiosmosis is in chloroplast, occurs during photophosphorylation, right? And the other differences are that in the chemiosmosis in mitochondria, electrons and protons are provided by reduced NADs and reduced FADs, right? Whereas in chemiosmosis in chloroplast, hydrogens and electrons are provided by the water. Hydrogens and electrons are released by the photolysis of water, right? And we can also say that in the chemiosmosis in mitochondria, the final acceptor of electrons and protons is oxygen, whereas in chemiosmosis in chloroplast, the final acceptor of electrons and protons is NADP, which makes reduced NADP. So in chemiosmosis in mitochondria, water is formed when oxygen picks up electrons and protons, whereas in chemiosmosis in chloroplast, reduced NADP is produced when NADP picks up electrons and protons. And the other differences are that in chemiosmosis in mitochondria, hydrogen ions are pumped from the matrix to intermembrane space, whereas in chemiosmosis in chloroplasts, hydrogen ions are pumped from the stroma to the thylakoid space. So guys, there are a lot of differences. We just need to write 3 points. So let's write them down. We can also say that chemiosmosis in mitochondria occurs in the inner membrane or cristae, whereas chemiosmosis in chloroplast occurs in the thylakoid membrane, right, or the membrane of the grana. Okay, let's write it down. the differences. In mitochondria, chemiosmosis occurs during oxidative phosphorylation, whereas in chloroplasts, it occurs during photophosphorylation. All right. The second difference is that in mitochondria, reduced NAD slash reduced FAD give protons and electrons, whereas in chloroplasts, these are made available or provided by photolysis of water, right? And what else can we write? Oxygen in mitochondria, oxygen is the final acceptor of protons and electrons to form water. In mitochondria, oxygen acts as the final acceptor of protons and electrons to form water, whereas in chloroplast, NADP acts as the final acceptor of protons and electrons to form reduced NADP. So guys, these are the 3 points that we can write. There are other points as well that we discussed earlier that we can also write. So we are done with question number 2. We will now move on to question number 3.